Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
Crosslinking and immunoprecipitation sequencing. Table of contents. Introduction. RNA-Protein Interactions. Principle. Steps. Types. Advantages. Limitations. Applications. Introduction. Crosslinking and immunoprecipitation. Sequencing is a method used to study RNA molecules and their interactions with associated proteins in cells. It is used to map RNA-protein interactions, which is useful for studying gene regulation. This is an antibody-based method that uses specific antibodies to isolate RNA-protein complexes. The RNA-protein complexes are cross-linked using ultraviolet light. RNA fragments are then extracted and sequenced. This helps identify the regulatory roles of RNA-binding proteins and their interactions with RNA. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Protein interactions. Proteins interact with RNA as soon as it is made from DNA during transcription. These proteins bind to the transcribed RNA to form ribonucleoprotein complexes and are called RNA-binding proteins. RBPs help regulate RNA processes like splicing, translation, transport, and overall function. They play an important role in regulating gene expression. It is important to construct an accurate map of protein-RNA interactions and study these interactions to understand gene regulation. Different methods have been developed to study protein-RNA interactions. There are protein-centric and RNA-centric methods. Protein-centric methods focus on a specific RNA-binding protein and identify all the RNA sites it binds to, helping to understand the role of that particular protein in gene regulation. RNA-centric methods look at all the proteins that bind to a particular RNA molecule and help understand how different proteins interact with RNA in the cell. Crosslinking and immunoprecipitation sequencing is a protein-centric method that provides accurate information on RNA-protein interactions and helps to understand RNA regulation. Principle. Crosslinking and immunoprecipitation. Sequencing works by using UV crosslinking and immunoprecipitation to covalently bind RNA molecules to associated RNA-binding proteins and isolate the RNA-protein complexes. The RNA fragments bound to RNA-binding proteins are extracted and sequenced to study the RNA-protein interactions. Initially, crosslinking is done by irradiating the cells with UV light, which forms an irreversible covalent bond between RNA and RNA-binding proteins in living cells. These linked RNA-protein complexes can be precisely isolated using immunoprecipitation. The complexes are purified to separate proteins and associated RNA fragments. After purification, RNA fragments are extracted, reverse transcribed, and sequenced using high-throughput methods. This process constructs a detailed map of RNA-protein interactions. Steps of crosslinking and immunoprecipitation sequencing. First, UV crosslinking. The first step in crosslinking and immunoprecipitation sequencing is crosslinking, which involves irradiating the sample with UV light. Exposing the cells to UV creates irreversible covalent bonds between RNA-binding proteins and their target RNA molecules. Most crosslinking and immunoprecipitation sequencing protocols use UVC light 254 nanometers, which can crosslink without requiring any pretreatment of cells. Cells are usually kept on ice for a short period during crosslinking to prevent UV-induced DNA damage. Second cell, lysis and RNA fragmentation. After crosslinking, the cells are lysed to release the RNA-protein complexes from the cells. The sample is then treated with limited amounts of RNases to fragment the RNA into smaller pieces. Fragmentation is done to generate RNA fragments of manageable size for later steps in the workflow. Third, immunoprecipitation. Immunoprecipitation is performed to isolate the RNA-protein complexes. The sample is incubated with antibodies that specifically recognize the RNA-binding protein, RBP, of interest. These antibodies bind to the target RBP, forming RNA-protein antibody complexes. To capture these complexes, protein beads or other affinity-based systems are used. After binding, the bead antibody complexes are thoroughly washed to eliminate any non-specific proteins or RNAs, ensuring that only the RNA-protein complexes specifically associated with the target RBP remain. Fourth, RNA isolation. The purified ribonucleoprotein particles are visualized using polyacrylamide gel electrophoresis. The resulting complexes are transferred onto a nitrocellulose membrane, and RNA molecules are visualized using radioactive labels. Then, the proteins are digested by proteinase K, and the attached RNA fragments are released. Fünfte, adapter ligation. Adapter ligation is necessary for reverse transcription and PCR amplification. In the original protocol, adapters are ligated to both ends of the RNA fragments after isolating RNA. However, this requires an additional purification step to remove the adapter. In newer variants of cross-linking and immunoprecipitation sequencing, only the reverse adapter is ligated to the 3' end of the RNA fragments, and this occurs after immunoprecipitation, while the RNA molecules are still in the complex. This is called on-bead ligation, and it removes the need for extra steps to remove the adapter. Another adapter is also ligated later, either during or after reverse transcription, depending on the specific method. 6. Library preparation. The isolated RNA fragments are converted into complementary DNA for sequencing. The resulting cDNA is purified to remove any unwanted byproducts. cDNA purification can be done using gel electrophoresis, which removes excess adapter sequences or primers that could interfere with downstream processes. However, gel electrophoresis can be time-consuming and labor-intensive. Other recent methods for purification include using silica beads or biotinylated adapters, which are quicker and more efficient alternatives to gel electrophoresis. Then, the purified cDNA is amplified using PCR, which creates a library ready for sequencing. 7th, sequencing. Finally, the cDNA library is sequenced using high-throughput sequencing methods. The generated sequencing data is processed and analyzed to identify the RNA-protein interaction regions. 8. Data analysis, crosslinking, and immunoprecipitation. Sequencing data analysis involves 4 main steps. First, pre-processing of sequencing reads is done to remove low-quality data and trim adapters. Then, the cleaned reads are mapped to a reference genome. The third step is peak calling, which identifies enriched binding sites as peaks. These peaks represent regions where the RNA-binding protein binds to the RNA. Computational methods help to separate these peaks from background noise in the data. Finally, post-processing analyzes these binding sites to identify motifs, gene locations, and biological functions. Types: high-throughput sequencing, crosslinking, and immunoprecipitation. One of the original crosslinking and immunoprecipitation sequencing versions, combining crosslinking and immunoprecipitation with next-generation sequencing. Uses UV crosslinking to create covalent bonds between RNA and proteins. Protein-RNA complexes are purified, and RNA is sequenced to map RNA-binding protein-binding sites. Photoactivatable ribonucleoside enhanced crosslinking and immunoprecipitation. Uses photoreactive nucleosides like 4-thioridine or 6-thioguanosine. Cells are treated with these modified nucleotides that create stronger and more specific crosslinking under UVA, light 365 nanometers. Causes mutations in sequencing data, allowing easier identification of exact protein-RNA interaction sites. Mutations from reverse transcription at cross-linking sites allow accurate mapping of binding sites. Use of artificial nucleotides may not work well in all cell types, and 4-thioridine can cause toxicity in some cells. Individual nucleotide resolution, cross-linking, and immunoprecipitation. Developed to overcome reverse transcription stops at cross-link sites causing truncated cDNA in high-throughput sequencing, cross-linking and immunoprecipitation, and photoactivatable ribonucleoside-enhanced cross-linking and immunoprecipitation. Attaches a 3' adapter to protein RNA complexes and adds the 5' adapter later during reverse transcription. cDNA is circularized and linearized, using a restriction enzyme to improve recovery of truncated fragments, preventing loss during library preparation. Enhanced cross-linking and immunoprecipitation involves 2 adapter ligation steps, a 3' adapter ligated to immunoprecipitated RNA, and a second single-stranded adapter added to cDNA after reverse transcription. Includes a size-matched input control to normalize background noise and reduce biases in data analysis. Infrared cross-linking and immunoprecipitation uses a 3-foot adapter labeled with an infrared fluorescent dye instead of radioactive labeling. Simplifies library preparation and improves efficiency. Requires fewer cells and has a faster workflow. Includes on-bead nucleus digestion for better RNA recovery and uses thermostable reverse transcriptase to minimize bias. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Advantages: cross-linking and immunoprecipitation. Sequencing can accurately capture RNA-protein interactions. Ultraviolet cross-linking in cross-linking and immunoprecipitation sequencing allows direct identification of RBP interactions with RNA. It does not cross-link proteins to each other, reducing background noise. Cross-linking and immunoprecipitation sequencing has broad applications in RNA research and is useful for studying processes like RNA splicing, RNA stability, and gene expression. The immunoprecipitation step provides high specificity in identifying RBP RNA interactions by using specific antibodies. This prevents nonspecific binding and makes the results more reliable. Crosslinking and immunoprecipitation's sequencing is a high-throughput method that can analyze a large number of RNA-protein interactions, making it useful for large-scale studies. Limitations: crosslinking and immunoprecipitation. Sequencing involves many complex steps like crosslinking, immunoprecipitation, RNA fragmentation, and reverse transcription, requiring specialized expertise and careful handling for accurate results. UV crosslinking has low efficiency compared to formaldehyde crosslinking, which can result in partial or inaccurate results and loss of some relevant data. UV crosslinking can also cause mutations in DNA. The method requires high-quality and specific antibodies for immunoprecipitation of specific RNA-binding proteins. Crosslinking and immunoprecipitation sequencing is less effective in detecting low-abundance interactions. It may be difficult to accurately map structurally complex or highly folded RNAs. The technique is expensive and labor-intensive, requiring specialized reagents and equipment, and takes longer time, making it less accessible for large-scale or high-throughput studies. RNA fragmentation and library preparation steps can introduce potential biases that affect the accuracy of sequencing results. Applications: crosslinking and immunoprecipitation. Sequencing is used to study RNA-protein interactions, helping to understand how gene expression is regulated after transcription. It helps to understand RNA processing and splicing by studying how RNA-binding proteins affect RNA splicing and identifying splicing factors. Crosslinking and immunoprecipitation sequencing is used to study RNA modifications and their functions in different cellular processes. It has applications in disease research by identifying altered RNA binding sites in disease states. Many neurodegenerative diseases and cancers involve dysregulated RNA-protein interactions, and crosslinking and immunoprecipitation sequencing helps identify disease-associated RNA-binding proteins and their target RNAs. The method is applied in plant research to study RNA regulation in plant development and identify RNA-binding proteins involved in disease resistance and genetic improvements. Crosslinking and immunoprecipitation sequencing can be used to develop RNA-targeted drugs, aiding in designing small molecules or RNA-based therapeutics for genetic disorders. Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you!